Hello everybody, welcome back to my channel Pharma. This is Devi Sarojini. In this video, I will discuss about the key points of analytical method validation. They are examples of methods that require validation documentation, published guidances for analytical method validation, types of analytical procedures to be validated. Next and final one, considerations prior to method validation. In this video, I will discuss about all these things one by one. I am giving main focus only on types of analytical procedures to be validated. They are like this: identification tests, quantitative tests for impurities content, limit tests for the control of impurities, quantitative tests of the active moiety in samples of drug, substance or drug product or other selected components in the drug product. Please watch full video without skipping for clear understanding of this topic. Let's discuss one by one. Examples of methods that require validation documentation are like this: chromatographic methods, spectrophotometric methods, capillary electrophoresis methods, particle size analysis methods, dissolution methods, titration methods, and finally automated analytical methods. These are all examples of methods that require validation documentation. These are all main examples of methods that require validation documentation in pharmaceutical laboratories: chromatographic methods, spectrophotometric methods, capillary electrophoresis methods, particle size analysis methods, dissolution methods, titration methods, and automated analytical methods. The published guidances of analytical method validation are like this: ICH-Q2A text on validation of analytical procedure in 1994, ICH-Q2B validation of analytical procedure methodology in 1995, CDER reviewer guidance. This is reviewer guidance validation of chromatographic method in 1994. CDER submitting samples and analysis data for method validations in 1987. CDER analytical procedure and method validation in 2000. CDER bioanalytical method validation for human studies. USP 1225 validation of compendial methods. These are all published guidances for analytical method validation. Next one, types of analytical procedures to be validated. I am giving main focus on this topic: identification tests, quantitative tests for impurities content, limit tests for the control of impurities, quantitative tests of the active moiety in samples of drug, substance or drug product or other selected components in the drug product. Let's discuss one by one. First one, identification tests. I will give you a clear explanation about identification tests. Identification tests are used to verify the presence of a specific element, functional group or compound, and can be biochemical, immunological or molecular biological. Here's a more detailed explanation. Identification tests encompass a wide range of techniques used to determine the identity of a substance or organism. They help verify the presence of specific components or characteristics like enzymes, proteins, or genetic sequences. Uh, Examples of identification tests are like this. Identification tests are intended to assure the identity of analyte in a sample. This is normally achieved by comparison of a property of the sample, that means spectrum, chromatographic behavior, chemical reactivity, etc., to that of a reference standard. These are the identification tests. Identification tests are a fundamental part of quality control, ensuring that the final product meets the required specification and contains the intended ingredients. Common identification tests are like this: chromatography, spectroscopy, elemental analysis, SIS, stability testing and particulate analysis. Chromatography: generally the techniques like HPLC, GC are used to separate and identify components in a sample. Next one, spectroscopy. Techniques like UV, visible, IR and NMR spectroscopy can provide information about the structure and identity of compounds. Next one, elemental analysis. It determines the elemental composition of a sample, which can help identify the presence of specific elements. Microbiological testing, also identification test. It identifies and counts microorganisms present in the product, ensuring it is free from harmful bacteria or fungal. Next one, essays. These tests measure the amount of the active ingredient or other components in a sample. Stability testing is also identification test. These tests determine how long a product can remain stable and effective under specific storage conditions. Next one, particulate analysis. It identify and quantify any particulate matter present in the product. These are all identification tests. Next one, quantitative tests. Quantitative tests determine the precise amount of a substance present in a sample, ensuring accurate doses, consistency and quality of drug product. What are quantitative tests? Quantitative tests are like this. These are analytical methods that measure the amount or concentration of a substance in a sample. Examples of quantitative tests: SI. It determines the exact amount of the active pharmaceutical ingredient in a drug product. Impurity analysis, measuring the levels of impurities and degradation products to ensure they are within acceptable limits. Next one, shelf life determination, monitoring the stability of the drug over time to determine its shelf life. Dissolution testing, analyzing how quickly and completely a drug dissolves in simulated body fluids. Next one, limit test. A limit test is a quantitative or semi-quantitative test that detects and limits small amount of impurities in a substance. Example is limit test for chloride. These are much more important because impurities can reduce the therapeutic effect of drug.